Good day everyone, I am Desert and Francisco, and together with me are Mr. Vivera and Mr. Olimpian, and today we will be presenting our post-laboratory results on the phytochemical screening of Cosmos bipinnatus, focusing particularly on its leaf. Procedures of plant extraction. For the extraction process, we started with 150 grams of fresh Cosmos bipinnatus leaves, which we dried for 3 days. Then the dried leaves were macerated with 80% ethanol in Erlenmeyer flask for 2 days. The mixture was filtered while hot, and the residue was washed again with ethanol, and filtrates were combined. We then evaporated the combined solution to dryness for about 12 hours, leaving the concentrated extract ready for testing. So as you can see, from 150 grams of fresh leaves, we obtained only 67 grams of dried material. The maceration process using 1 liter of 80% ethanol gave us 504 ml of filtrate. After heating to dryness for around 12 hours, we collected a total of 4.69 grams of the plant extracts, and these were our actual laboratory results. For the organoleptic evaluation, the extract appeared to be deep brownish green in color, dry and flaky in texture, and it was in solid form, and these were the actual characteristics that we observed during the experiment. For the percentage yield, the empty beaker weighed 36.77 grams, while the beaker with the plant sample weighed 100.77 grams. This means that our plant sample was 64 grams and the extract was 4.69 grams. The equivalent plant material per gram of extract was calculated as 13.65 grams, and these were the actual results derived from our experiments. For the chemical test, first is alkaloid detection. So we included Mayer's, Wigner's, Jagendorf, and Hager's test. However, these results were only theoretical. We did not perform these tests in the laboratory, and based on theoretical interpretation, all tests showed negative results indicating that alkaloids are absent in Cosmos bipinnatus. For the carbohydrate detection, we used Mollick's, Benedict's, and Felling's tests. However, the results of these tests were only theoretical and we didn't actually perform them in the lab. So all tests showed positive results with orange to red precipitate, confirming that the reducing sugars are present in the extract. For the detection of glycosides, both modified born trader tests and legal tests were theoretical results only and were not performed in the lab. The theoretical results indicated that there is no observable color change, meaning anthranol and cardiac glycosides are absent in the extract. For saponins, we performed the froth and foam test in the laboratory, and both gave positive results, showing foam layers that lasted for around 32 minutes, and this confirmed the presence of saponins in the extracts, which are known for their foaming and medicinal properties. For phytosterols, both Salkowski and Lieberman-Burchard tests were theoretical only. Theoretical results indicated that there is no color or ring formation, suggesting that phytosterols are absent in the Cosmos bipinnatus leaf. For phenolic compounds, all 4 tests including ferric chloride, lead acetate, alkaline reagent, and magnesium HCl reduction were only theoretical results. These theoretical findings indicated positive reactions such as bluish to black coloration and precipitate formation, suggesting the presence of phenolic and flavonoid type compounds in the extract. Next would be the detection of tannins. So the gelatin test for tannins was again theoretical only. The theoretical results showed a white precipitate indicating that tannins are present in a very small amounts in the leaf extract. The centroproteic, ninhydrin, Milons, and Biret tests were used for detection of protein and amino acids. However, centroproteic test was the only test that was performed during the laboratory activity. All others are theoretical. And the color reactions including yellow, blue, red, and violet changes suggest that the proteins and amino acids are present in the Cosmos bipinnatus plant. The copper acetate test for diterpenes was theoretical. The emerald green color change theoretically indicates that diterpenes are present, which are compounds essential for plant growth regulation. For fixed oils and volatile oils, both the stain and saponification tests were theoretical. The theoretical results that suggest that there are no fixed oils present, but a small amount of volatile oils may exist in the extract. To summarize our findings, based on actual lab work, we confirmed the presence of carbohydrates and saponins and proteins. The rest, including alkaloids, glycosides, and phytosterols, were theoretically determined to be absent, and these results suggested that the Cosmos bipinnatus leaves have some active compounds responsible for biological activity. Additionally, from the theoretical analysis, we also found the presence of phenols, flavonoids, proteins, amino acids, diterpenes, and small amounts of tannins and volatile oils. Fixed oils were absent, and overall, combining both our actual and theoretical results, the Cosmos bipinnatus leaf extract shows several bioactive compounds that may contribute to its antioxidant and medicinal properties.
Moving on to the answers to questions part. Number 1, what is phytochemical screening of plant constituents? Phytochemical screening is a laboratory method used to identify the chemical compounds found in plant extracts. So it detects groups of bioactive substances like alkaloids, flavonoids, tannins, saponins, steroids, and glycosides through specific chemical tests that produce color change or precipitates. So this process provides qualitative information about the plant's constituents without determining their exact quantities. Number 2, what is the purpose of preliminary plants constituents? So the purpose of preliminary phytochemical screening is to identify the main bioactive compounds in plants that may have medicinal value. So it helps pharmacists and researchers recognize which plant extract could lead to potential drug development. So it also provides a scientific basis for the traditional use of medicinal plants and guides further pharmacological and chemical analysis. Number 3, why is 80% alcohol the recommended solvent for the preparation of plant extract for preliminary screening? So 80% alcohol is recommended for plant extraction because it can dissolve both polar and non-polar compounds. So the water helps swell plant tissues for better penetration, while the alcohol extracts substances like alkaloids, flavonoids, and phenolics that water alone cannot dissolve. So this balanced mixture improves extraction efficiency and yields more bioactive constituents, making it ideal for phytochemical screening of Cosmos bipanatus leaves.
The analysis of plant extract for phytochemical indicate the presence of saponins, phenolic compounds, and proteins. The foam and froth tests both produce a stable foam layer confirming the presence of saponins, while the ferric chloride tests indicate the phenolic compounds to the bluish-black coloration. The xanthroprotic test yields a yellow coloration signifying the protein contents. Overall, it yields a positive result and confirms that plant extract contain multiple bioactive constituents, demonstrating its potential pharmacological and biological value.